special case of part engineering, along with promoter engineering, RNA engineering. So I don't view it as an orthogonal thing, but it's a narrower subject than synthetic biology is.
So there's no sort of greater, or is there? Are you trying to make this like a medicinal therapeutic?
No, it's an application directed towards a foundational problem. So it is not a direct real-world application. It is not trying to cure cancer with sound-induced self-lysing E. coli. It's nothing like that. And it's certainly not for making biofuels directly. But it's a foundational technology, and making bacteria that can cure cancer or making biofuels are both technologies that would be very much advanced by having better foundational technologies. So, if our team is successful in developing bacteria that really only need sound and heat in terms of the processing you're doing to the cells to get standard assembly reactions to happen, then it's going to be really cheap and easy to do standard assembly reactions. And that means we're going to be able to access more composite parts, and we're going to get a lot more data about functional composability of basic parts into devices. And from that, we're going to be able to do problems like biofuel bacteria or therapeutics a lot faster than we could before. So that's the application of it. I mean, in general, the theme here is using synthetic biology to solve foundational problems in synthetic biology.
And how does this differ from last year's IGEM?
Last year we did an application that was so last year's project was to make Bacto blood. The idea was to have a bacterial-derived red blood cell substitute derived from E. coli. And the interesting thing about that project was it was the first attempt to make a system in IGEM. So it was a bacterium that had many things going on at once. It was making heme, which is a small molecule. It was making the proteins that were needed to make hemoglobin. It was also making proteins that would allow them to be freeze-dried. And it kind of coordinated it all together with this controller device that was, it changed copy number. It was, it was this big elaborate system with many things going on at once. And so that was the real focus of that project, was figuring out exactly what it means to make a system in synthetic biology, because the extreme majority of research in synthetic biology has all been about making devices, or not even getting devices, just making parts.
And so once again, that was a functionality. It was learning how to make a system versus making something.
I would say, yeah, that's true. Even the applications we do are still kind of foundational in the sense that, whenever you're approaching a new application area that is something distinctly harder to do than what has been done before, you can't necessarily approach it with the goal of having a successful product. I mean, a lot of it is just figuring out what's it like to try to make something like that. And you figure out that, we don't necessarily have all the tools to make that thing. So a lot of what I do, at least in terms of the applications that we pick, is we try to pick these big applications that we know we can't easily solve. But just the act of trying to do it, we'll figure out what are the problems with making that sort of thing. And the problems we identify then become the targets for our foundational technology efforts. So, and then we solve those foundational technologies, and those will help these projects more generally move forward.
Perfect. And so how did you come about choosing this year's IGEM project?
So this year's project was a combination of things. So we had asked the students what they wanted to work on, and the idea of doing a sound sensor was proposed by 3 of the students involved in it this year. So, and that you know, we have a steering committee of faculty people who kind of help pick the team and deal with the financing and the location of the room and all that jazz. And that is not something that any of us had thought of even trying to do. That was totally student-derived. But really, we felt that was not a big enough goal for IGEM. We like to think a little bigger than that. You know, you want to have kind of short, easy goals, intermediate goals, and like, you know, grandiose scheme goals as well as part of these things. And hopefully you at least get the small goal made and maybe the middle goal and progress on the big goal. So another student, though, had also proposed this idea of having bacteria, in effect, make the reagents to do standard assembly. And didn't really have a scheme in mind for how to do that. But, you know, we kind of had this idea on one hand of sound. We had this idea of making reagents, so making the restriction enzymes in the E. coli. And then just someone in my lab who ended up being the TA for IGEM was developing these anti-holin lysozyme type circuits for a totally different application than this. And so kind of all those ideas were buzzing around, and they seemed to fit together into this kind of interesting project. So there were a lot of discussions between the students and the TAs and the instructors, and we considered other things to do. I mean, the idea for sound-induced self-lysis came pretty early. We just kind of liked that idea, just that as a device in and of itself seemed like a cool thing, and there had to be applications for something like that. But we considered a lot of different applications before kind of deciding on these three things we were going to try to do derived from it.
And how long have the students that are on the IGEM team now, when did they start? Like, when were they involved in this decision process?
So, all right, so we kind of have a long drawn out process here that starts, this past year we started around March when we had the first applications, and the first applications were just like 100 words, why do you want to do IGEM? And then we invited all those people to come to a meeting where we told them a little bit what the program was about, tried to get a few of them to not want to be part of IGEM anymore. So we started with about 50 people, and we whittled it down to about 20 people after that meeting. And then those 20 people were each asked to do a series of online tutorials, all directed towards how you make basic parts. And also within that were questions about, name 3 project ideas for IGEM. And then from that list of 20, we kind of whittled down further based on their performance on the tutorials, their ideas, and just kind of their interest areas. Wanted to see if they really wanted to do synthetic biology based on kind of the ideas they were proposing, and the degree to which they understood what they were getting into. And then finally we did interviews. And from those interviews we finally whittled it down to the 5 core members of the team, plus the 2 high schoolers that ended up on the team, and the high school teacher that ended up on the team. And so they showed up. Well, okay, so then they finished the rest of the tutorials, and then June 2nd they showed up in the lab, and we began the training process, which consisted of a series of lectures that largely recapped what was in the online tutorials, but there's something about just saying it versus reading it. So we redid a lot of that and went into a lot deeper detail about specifically how to do things, specifically how to keep records of what you're doing. We had more group discussions about the project and started parsing out the project into individual goal projects. And then they started work. They started making basic parts. Mm-hmm.
One last question. What sort of lessons do you think you learned from doing IGEM these past 2 years that that?
One of the great things about IGEM is it lets everyone